Hi, everyone. My name is Kaitlyn Ford, and I'm a postbac research intern in the lab of Dr. Lucia Peixoto at Washington State University in Spokane, Washington. Today, I will be talking with you about my poster titled Global Transcriptional Profiling of the Effects of Sleep Deprivation at the Gene, Transcript, and Single Nuclear Level. Sleep deprivation has been shown to affect cortical gene expression, and Alexander Barbet found that sleep is important for normal brain function. Homer1, a scaffolding protein that I will discuss later, has an isoform lacking the carboxy terminal domain that has previously been shown to be upregulated by sleep deprivation. This transcript is Homer1A. Other Homer1 transcripts are constitutively expressed. Given that sleep deprivation affects cortical gene expression, we investigated these effects at the gene, transcript, and single nuclear level in wild-type mice. We began by separating mice into two conditions: those who were allowed to sleep, also referred to as our home cage samples, and those who were sleep deprived. We collected prefrontal cortex tissue from both, and then went on to do both RNA-seq and single nuclear RNA-seq. For our RNA-seq samples, we had replicates of 5, each with 30 to 50 million 100 base pair end breeds. For our single nuclear RNA-seq samples, we had replicates of 3, each with 10,000 nuclei, and we had 30 to 50,000 100 base pair end reads per nuclei. Our RNA-seq data was quantified using salmon quant. We performed upper quartile normalization using the E2seq package. We estimated batch effects using RUV-seq and had a k-means clustering of 3 at the transcript and gene level. We used the swish method for fish pond for differential gene expression, differential transcript expression, and differential transcript usage analysis. Differential transcript usage looked at how the proportions of transcripts changed across conditions. Our single nuclear RNA-seq data was quantified using salmon albumin. We began by adding exons and introns and adding a count of 1 to the mitochondrial counts. We used Skater and Scuttle for quality control. Scuttle was used to detect low quality and damaged droplets. We used the fast MNN method from Bachelor to correct data from batch effects. We used single R as the annotation for our data. This was from the Brain Initiative Cell Census Network, and we did this to avoid bias from clustering. Downstream analysis included the negative binomial generalized model from EdgeR and the negative binomial mixed effects model from Muscat. First, I will talk about the differential transcript expression and differential gene expression data. We found that sleep deprivation downregulates transcripts that are not detected at the gene level. Here on the left, you can see our PCA plots. You can see a separation of our home cage samples in green. These are the animals that were allowed to sleep. You can see along the first principal component, they are separated from our sleep depth samples in orange. Overall, we found over 7,000 significant genes and almost 9,000 significant transcripts. These are shown in blue on our MA plots, which are plots B and plots D. We elected to highlight Homer1, MAPK3, also known as ERK1, FOS, and ARK, which are shown in shades of red. FOS and ARK have activity-dependent gene expression and have previously been seen to be upregulated by sleep deprivation. We found out almost an 80% of recovery of positive controls at the gene level from a prior established data set. And one thing that I would like to point out is that we see this downward tail at the transcript level, but we do not see it here at the gene level. Moving on to our differential transcript usage data, we found that the proportion of Homer1A increased while Homer1C and Homer1D decreased. We found over 1,700 significant transcripts with a Q-value less than 0.05. And you can see here on our MA plot, Homer1A has a positive fold change, while Homer1C and Homer1D have a negative fold change. We found that sleep deprivation has a large detectable effect at the single nuclear level. Here, you can see a multidimensional scaling plot. Each point represents a biological replicate. Our circles are animals who were sleep deprived, and our triangles are the animals who were allowed to sleep. Not only can you see a separation between the circles and the triangles, or the animals that were allowed to sleep and those who were sleep deprived, but you can see a separation of excitatory, which are glutamatergic neurons, and inhibitory, GABAergic neurons. Over here in our key, you can see we have the glutamatergic neurons, which are over here selected in purple, and our GABAergic neurons, which are green. On the plot, our glutamatergic neurons are here, while our GABAergic neurons are here. When we looked at gene expression at the single nuclear level, we found FOS and HOMR1 across both glutamatergic and GABAergic cell types. To conclude, our differential transcript expression analysis allowed us to detect downregulated transcripts that were not detected at the gene level. Our differential transcript usage analysis showed an increase in the proportion of HOMR1A and decrease in other HOMR1 transcripts in response to sleep deprivation. We found that there was a large detectable effect by sleep deprivation at the single nuclear level. Thank you so much for listening. I would like to acknowledge everyone that worked on this project and especially highlight those whose names are bolded on the right. Thank you.